First of all, my sincere thanks to the organizing team for giving me this opportunity for this unique and learning experience. At least for me, drug elution is a learning experience. Though I am the opening batsman for this meeting, but my position is exactly like the player sitting on the bench, watching and learning Sachin Tendulkar as the opening batsman. Historical development is always very fascinating. The farther backwards you can look, the farther forwards you are likely to see. And lesson learns from historical evaluation, the key to rise, rule, road towards a progressive and permissive future. If you know the history, I think will not rukega nahi Bharat. Well, any endovascular meeting is incomplete mentioning the name of two giants who basically fundamentally started the endovascular interventions. The two giants, Daughter and Grundgich. We should also thank and not to forget the first patient who submitted herself for the first endovascular procedure. This is the story of Laura Shaw, the beginning of endovascular era. She was 82-year-old lady, suffered with rest pain, left foot with gangrenous toes, advised amputation by all. She refused. And subsequently, she was referred to Charles' Daughter. He said he will do an angiogram first and then probably he'll take a call. He did an angiogram and then he found out there is a SFA lesion, short segment SFA lesion, and he planned for dilatation of the lesion. And that is how the story started. This is the story of Laura Shaw, the first peripheral angioplasty done on January 16th, 1964 at 2 p.m. You can see the result, fantastic flow started, and the notes written by Daughter himself. Happy lady, watching her angiogram, the success story, and she was relieved of pain and gangrene. He believed if a plumber can do it to pipe, we can do it to blood vessels. Very determined. Before that, two important events in 1963 preceded this first angioplasty. While he was studying coronary imaging in cadavers, he realized that the forceful injection across diseased arteries could disrupt and expand obstructing lesion. He reasoned that it should be therefore be possible to dilate similar lesions in the leg arteries. And the second event, performing an abdominal aortogram to specifically assess the renal arteries. When he eventually advanced his catheter through a occluded iliac artery, this set him and wonder about the possibility that had a balloon been attached to the catheter. He had then association with Cook, we all know, Cook industry, and they both sat together and decided to have more and more work in this endovascular arena. He was unstoppable. Not only diagnostic, he started doing all therapeutic work also, and so much so that the surgeons became upset. He started taking cases from the surgeon. So now, very specifically, surgeon used to write in their prescription, visualize, but do not fix it. We are there to fix. But not all cases of Daughter did well in the follow-up. The reason? It was longitudinal shear force along the axis of the artery that created the problem. The solution? It would be better if all the force could be directed radially outward and only minimal or no longitudinal shear state at all. And so, the concept of idea of a balloon. Andreas Grundgich, the gentleman who brought the concept of balloon angioplasty. A balloon attached to tip of a very thin flexible catheter could be introduced into the vessel via a small puncture hole, could be maneuvered around curves, bends, could be passed through an obstruction with a minimal shear force, and once placed into position could be inflated to a larger size, applying only radial pressure and dilating the channel. The first balloon catheter constructed by him in 1971, and the first patient whom he did a peripheral balloon angioplasty was Fridge Ott, that was done in February 1974. Another photograph of balloon dilating catheter handmade by him which was used for first iliac balloon angioplasty on March 6, 1974. Double lumen catheter for iliac balloon angioplasty. Again, one more case of SFA lesion and dilating it. The landmark story of him is that September 16th, 1977, the first coronary angioplasty was done by him. You can see his patient sitting in front of him on his favorite Vespa scooter. Master at work, both of them received a nomination for the Nobel Prize in Physiology and Medicine 1978, became one of the most successful examples of translation medicines into the 20th century. Saved more than 1.5 million lives, prevented millions and millions of amputations. 1985, the black year in the endovascular era. Both of them, we lost both of them together in 1985. The story continued. Angioplasty, balloon, catheter, we always had a problem, recoil. What was required was a mechanical support. And then people started venturing more and more. They got an idea that steel arches used to stabilize tunnel construction maybe can be used for the arteries as well, and why not a stent? So the era of stenting started. The name stent, Charles Thomas Stent, an English dentist, and that is how the name of a stent. 1980, the two gentlemen, Swedish gentlemen, they met and they had a project, Singh, a cardiothoracic surgeon, and a Hans Wallsten. Both of them, with the help of engineer Christian Imbart, they eventually developed the first stent for the use in coronary arteries, that was the wall stent. The first implanted peripheral stent graft in 1985, Rojuos. March 1986, Jacques Piol implanted the first coronary stent graft. Very important picture. The first endocoronary stent was implanted in the men during the month of March 1986. And we see the result after 18 years of follow-up, the stent is patent. Same time across the globe, Palmer, he was there, he was very much fascinated by this concept, and he also started working, and they developed, with the help of his friend, developed a Palmer stent. The first stents were made from stainless steel, and despite their thick studs, poor flexibility showed superiority over POVA. With elimination of abrupt occlusion and a reduced rate of restenosis, confirmed in two historical trials published in 1993, Benny stent and stress trial. Ideal stent. An ideal stent should have a good flexibility, deliverability, low thrombogenicity, strong radial force, good biocompatibility. And this is how a stent structure looks like. We have studs, rings, cells, crowns, and connectors. But in real scenario, in spite of stent, we have a major issue. The Achilles heel, that is restenosis and in-stent stenosis. The problem remains neointimal hyperplasia. Pathophysiology of in-stent restenosis and stent thrombosis, I think all of us know this. Attempt was made to cut down these kind of restenosis or neointimal neoplasia by cutting balloon with the help of heparin-coated stent, with the help of antibiotic-coated stent, but not of much use. And then the concept of drug elution, a promising toy in the millennium, has come. Drug is eluded by either intraluminal drug delivery devices or by an extraluminal drug device. Drug eluting stents work by combining both mechanical support of a traditional stent and with a drug delivery system. I'm sure the subsequent speakers are going to speak more on this. The stent scaffold keeps the artery open while the drug helps prevent restenosis by inhibiting excessive tissue growth, inflammation that can occur in response to stent placement. However, does have limitations, including the potential for late instant thrombosis requiring dual antiplatelet therapy. Drug-eluted stents coated with drugs to prevent restenosis There are various devices now. We have drug-coated stent, we have brachytherapy catheters, we have balloons, delivery balloons, we have drug-coated balloons, we have drug delivery capsules, we have drug-eluting spheres or beads, drug-eluting films or coating, drug delivery nanoparticles, drug-eluting sponge or gel, drug-eluting microcapsules. But the success of a drug delivery devices depends on tissue binding and clearance. Many factors collectively contribute to the complex interplay between vascular atherosclerotic structure and drug retention, highlighting the importance of considering plaque characteristic in drug delivery strategies for treatment of atherosclerosis. Plaque structures and vascular systems, a lot of factors, they depends upon the drug release and drug retention, endothelial permeability, plaque composition, vascular architecture, macrophage infiltration, neovascularization, calcification, lipid core size and stability, plaque erosion rupture susceptibility, collagen content, hemodynamic factor, and matrix metaproteinolysis. Well, the concept of DES in 1999, Sosa implanted the first DES in Brazil, signaling the third revolutionary shift in the history of interventional procedures. The first generation drug-coating stents, the drug-eluting stents were introduced in 2002 when Cordis Johnson & Johnson received a CE mark for Cypher, sirolimus eluting stent. Next year, Boston Scientific received a CE mark for another DES named Texas Express Paclitaxel, launched in 2004. These two stents make the first generation of stent and considered the pillar of DES technology. The initial enthusiasm was shaken in 2006 when Kamenjit published a meta-analysis showing the increased risk of death and myocardial infarction related to late and very late stent thrombosis, possibly related to delayed endothelization, secondary to anti-proliferative drug elution, hypersensitivity reaction to the polymer coating, thick struts, low suitability of the metallic platform. The issues of first generation steered the wheel of research group towards better and improved product, which resulted in introduction of stent with thinner strut, better polymer coating, and new drugs. Second generations. The benefits of everolimus includes better bioavailability, less clearance time, higher cellular residence time, good absorption as compared to sirolimus. Second generation stents gave a very promising result. However, the long-term clinical trial indicated the issue of late thrombosis, very late stent thrombosis, delayed healing, and inflammation at the later stage. So, third generation stents. The third generation stent were introduced enclosing stents with either coated with biodegradable polymer or stent without polymers, known as polymer-free stents. The fourth generation bioresorbable stents. The fourth generation, instead of a metallic platform, scientists developed a stent with a polymeric backbone that would disappear once the drug is eluted. This was a unique and creative idea, and many products came into the market, but none of them could stay on the market because of the issues. Their major issues was low mechanical strength and the drug release profile. This class is very attractive and has a potential, but still under research. They are designed to dissolve and absorbed by the body gradually over time. They are typically made of materials such as polylactic acid or magnesium alloy. The potential advantages include a reduced inflammatory response, lower restenosis rate, and ability to maintain normal vasomotor tone. They are suitable for pediatric patients and those with metal allergy. There are many trials regarding the effectiveness of these DES. I think subsequent speakers are going to discuss all these trials. Drug-eluting balloons, initially developed in 1980, experiencing a renewed interest as an alternative to address the limitation of drug-eluting stent. The primary limitation of DES lies in inconsistent drug delivery with only 15% of the vessel wall in contact with stent surface, hindering uniform drug elution. Again, there are many trials for DEB. I think we will be knowing about this more in the subsequent lectures. Porous, microporous balloons. Now, a word about extraluminal drug delivery devices, perivascular biomaterial. Extraluminal drug Delivery devices and perivascular biomaterials are designed to deliver medications around blood vessels rather than directly into them. Collagen wrap, mesh devices, drug delivery catheters, drug eluting hydrogel, then we have patches, coatings, scaffold, gels. Lastly, local drug delivery has become a prominent strategy for preventing restenosis after interventions for obstructive atherosclerosis. The success of locally administered drug is closely tied to the vessel's anatomy and morphology of the lesion being treated. As our understanding of lesion-specific therapies continues to evolve, there is a potential for expanded clinical application of local drug delivery devices. Lesson from history and ongoing advancement in the field provides a hope for improved outcome in treating vascular diseases. Historical developments are footsteps for future developments. Friends, there are more than 200 Taj hotels, but the first historical iconic the Taj Mumbai built on 16 December 1903 was the road towards a progressive chain of Taj hotels. The basis of growth of modern invention is science, and the science is almost wholly out growth of pleasurable intellectual curiosity. Thank you very much.